Hello and welcome back to educator.com and biochemistry. So today we're going to continue our discussion of membrane lipids by discussing the other classes that we did not discuss in the previous lesson. So let's get started. Okay, now I know that I mentioned 5 classes, and one of those classes was going to be the tetraether lipids. I've decided actually not to discuss those. No particular reason why. You're welcome to look them up in your book. Should be no more than a 5-minute reading. I decided to actually move on to what I would consider a little bit more important. So the next class I'm going to talk about are going to be the sphingolipids. So sphingolipids. Now, the sphingolipids are characterized. So instead of glycerol, the sphingolipids, actually, let me start again. So instead of glycerol, this thing called sphingosine forms the backbone of these sphingolipids, backbone to which the groups are attached, to which various groups are attached. Okay, so let's go ahead and draw the sphingosine structure. I am not doing very well at all with spelling today. Sphingosine structure. Okay, so we've got, yeah, that's fine. I'll go ahead and do it here. C, C, C. So we have an OH, and then we have an NH2, and then we have an OH, and then we've got C, double bond C. Let me go ahead and put the H's in here. Let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Let me see. I've got 1, 2, 3, 4, 5, 6, and yes, there we go. So this is sphingosine. Now, what's important to notice is that, so one of the nonpolar chains is part of the sphingosine itself. Is part of the sphingosine itself. So in some sense, it's kind of like the glycerol. I mean, you have the OH and you have the OH, but it isn't the OH that's attached. It's actually directly attached, the 1 and the 2 carbon and alkene linkage, and then of course you have this other set. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 carbons there. So this basic structure is sphingosine, and to this, so this will always be the same. This is invariable. It's the attachments that go here and here that changes everything. Okay. Now, when a fatty acid is attached to the nitrogen in amide linkage or an amide linkage, I actually prefer to say amide myself, amide linkage. In other words, when a fatty acid is attached to that, go back to black, the compound is called a ceramide or a ceramide. So let's go ahead and draw that real quickly. So we have C, C, C. So if I have NH and some R1 group. And of course, here we have, let me see, I'll do CH212 and just write it as CH3 instead of doing the squiggles like that. OH, and of course we have our OH. So this is a ceramide, where this can be variable. And again, notice this top structure right there, it's the same. That's the sphingosine. So this basic structure right here, that is going to be, that's the sphingosine. When a particular fatty acid is attached to the nitrogen, and this is your amide linkage, an amide linkage, instead of the carboxyl, instead of an ester, instead of this being an oxygen, this is a nitrogen, it's called an amide, carbonyl. So, okay, so now let's go ahead and talk about the subclasses. The subclasses of the sphingolipids. So we have these membrane lipids, and one of the classes is the sphingolipids. Among the sphingolipids, there are subclasses, so now we're going to divide it up some more. These are derived from the ceramides. So, derived from the ceramides. In other words, in the case of these particular subclasses that we're going to discuss, they're always going to have the sphingosine. They're always going to have some fatty acid attached in amide linkage to the amine group, and then there's going to be something else that's going to be attached here. So they are derived from the ceramides and differ in what is attached to the number 1 carbon. So let's go ahead and call this our number 1 carbon. Okay, so the first is something called the sphingomyelins. The second is something called the neutral glycolipids. And then, of course, number 3, we have something called the gangliosides. Oops, write it a little bit better. The gangliosides. Okay, so the first one we're going to be talking about, I'll go ahead and go to red. We're going to be talking about the sphingomyelins. Now, these contain phosphocholine or phosphoethanolamine, just like the glycerophospholipids. Phosphoethanolamine. Ethanolamine as the head group. Okay, so let's go ahead and do an example of this. So let's go ahead and draw it all out very carefully. So we have C, C, and C. We have C double bond C. We have a CH2 group. We have 12 of them. And then we have a, we have a CH3 group. We have our OH. Okay, we have our nitrogen. We have our amide linkage. I'll go ahead and just put R1 for whatever fatty acid happens to have reacted. And I'll go ahead and do an O here. And now it says there's a phospho group. So the sphingomyelins, they do contain phosphate just like the glycerophospholipids. So a glycerophospholipid and a sphingolipid, they're both phospholipids. They both contain phosphate. So go ahead and do that. And we said it was choline, so we have 2 C's. We have a nitrogen. And let me go ahead and actually draw in my CH3 groups. And let's look out for charge here. So we have a positive there. We have a negative there. So this right there. That's our choline group, and again, it could be ethanolamine. So that's it. So this is a sphingomyelin. It happens to have a phosphate group, a choline or an ethanolamine right there on the left. So this is the head group. And then, of course, we have part of the chain that is always going to be there. That's part of the sphingosine backbone. And, of course, this variable fatty acid that's connected to the amine in amide linkage. There you go. This is a sphingomyelin. Okay, so now, because, you know what, I'm not going to actually write all this out. It's going to take too much time. So again, what's interesting here is that phosphate shows up in these sphingomyelins. So we had glycerophospholipids. If we want, we can call them sphingophospholipids. That's how I differentiate them. They're phospholipids in general, but one of them, the backbone, is glycerol. The other one, the backbone, is sphingosine, or in this case, the ceramide. That's it. So again, I personally think that there's too much nomenclature in biochemistry. I understand that we have to, name this and name that. I have no problems with that. But I think there's definitely a little bit of overclassification. I think that if you just draw out the structure and deal with a particular molecule as you deal with it, I think that's enough. So again, as far as you learning this stuff for the first time, it's really, really important not to get bogged down in the classification. Yes, of course, I understand if you happen to have a teacher that wants to make sure that you can put a particular molecule in a particular class, in a particular subclass, that's fine. It all depends on what it is that your teacher wants. But what's important is that you understand the chemistry, not necessarily the classification scheme. In general, if you stop and sort of look at the name, it'll tell you what's going on. But don't get lost in it. You want to be able to see the forest from the trees. It's the chemistry that's important, not these classification schemes. They've always made me crazy, but you know what? That's the nature of the game. We live with it. So anyway, so let's go ahead and see if we can move forward here. Now let's go ahead and talk about the glycosphingolipids. Okay. Let me see here. Yes. Okay. So we've just talked about the sphingomyelins, and now we're going to be talking about the glycolipids. Okay. So let me do this in blue, or what I just wrote down earlier as the neutral glycolipids. So glycosphingolipid. Okay. Now, a couple of things. These occur mostly in the outer leaflet. Of a membrane bilayer. And they have one or more, usually not more than about 3 or 4, but so one or more sugar monomers. So I don't want you to think of like 15. It's not going to be like that. Sugar monomers attached to the ceramide backbone, attached to the ceramide without a phosphate linkage. So that's what's important. In the sphingomyelins, you have this phosphate group that's going to, it's going to be connected to the ceramide, and it's going to be connected to the particular alcohol or whatever it is that's attached. In the case of these glycosphingolipids, the sugar monomers are going to be directly attached without this phosphate intermediary. Okay, so let's go ahead and, so we have 2 types. Again, I personally think all of this extra classification is completely unnecessary, and it's just, I think it complicates matters. So I hope that I can make it as clear as possible. I think too much classification really is not a good thing. But you know, the material is presented like this, so we'll go with what they have. So our first type is going to be the cerebrosides. There's one monomer, one monomer, and that monomer is either going to be glucose or galactose. You see the galactose tends to show up in a lot of these. And the other, they are called the globosides. That's exactly what you think it is. They're the ones that have 2 or more monomers. And again, you probably won't see more than 3 or 4. 2 or more monomers. Okay. And last bit of information, both are electrically neutral. Electrically neutral. Okay, so let's go ahead and take a look at a cerebroside. So let's go ahead and do this one in black. So we have C, we have C, we have C, double bond C. I've got my methylene group, 12 of them. I have CH3. I have my nitrogen. I have my carbon double bond. I'll go ahead and put R1. And here, let's go ahead, I'll go this way. I'll go O. I'll go that way. Maybe not. That's fine. I don't know. It doesn't really matter. Let's go ahead and let's do it like this. Let's go ahead and do O, and let's go like that. How's that? And we'll go ahead and do something like this. Let's go OH. Let's go OH. Let's go ahead and put glucose on there today. CH2OH. This is a cerebroside. A cerebroside. It has one monomer of glucose or galactose attached to the ceramide, and this is usually what's going to be variable. That's it. It's called a cerebroside. Just happens to be one of the names for it. Okay, let's go ahead and do a globoside, and let's use 2 monomers. So we've got, again, our C, oops, I think you know, I always forget that OH there. My apologies. Yes, it's very, very important to remember that there is that OH there. So let me go ahead and put that in first. That's that. And we have our double bond. Yes. And then we have CH2, 12, CH3. And you can see how easy it is to get lost here. And it's N and C and R1. Let's go ahead and go O. Okay, let's go ahead and put one monomer here like that. And let's go ahead and connect this one this way. Like that. We'll put an OH. An OH, this looks like a glucose monomer, so this is CH2OH. Let's go ahead and make this one a galactose. So we have down, we have up, and we have CH2OH. So this is a globoside. That's it. Same thing. You have your ceramide backbone, you have a monomer, you have a monomer. In this case, we have glucose, and we have galactose. That's it. Neutral glycolipid. Neutral glyco is the sugar part. Sphingolipid. Glycolipid, glycosphingolipid, sphingoglycolipid. Again, names. Structures are what matter. Okay, now let's go ahead and go to our third subclass, which is the gangliosides. Very, very important. Not that the others are not, but these are, well, anyway, you'll see. Gangliosides. Okay, so these have oligosaccharides, as their head group. And one or more residues, this is the important part, of new 5AC Remember that? N-acetylneurominic acid, sialic acid. I'm just going to go ahead and call it sialic acid. Sialic acid at the terminal positions. So in this particular case, the terminal positions. Let's see. Yes. So the gangliosides, what separates them from something else, from the other, glycolipids, they have oligosaccharides as their head group, and they have one or more residues of this sialic acid at the terminal position, at some terminal position. In other words, it ends with some sialic acid residue, 1, 2, 3, however many. So, now, recall that sialic acid has, actually I'll write it as, recall that NEU5AC has a -1 charge at physiological pH. Okay, so gangliosides carry negative charge. Okay. Now they are broken down into series. Again, just more classifications that make you crazy. I swear, that just drives me insane. So the GM series, there's one sialic acid residue. So if you happen to have a ganglioside that has only one sialic acid residue, it's part of something called the GM series. So that's it. GM1, GM2, GM3, GM4. The M stands for monosialic acid. Ganglioside, monosialic acid residue. So when I have a molecule like GM1, GM2, GM3, that 1, 2, 3, that's part of the name of the molecule, the specific molecule, the specific ganglioside. The M is telling me that that molecule, whatever the particular arrangement of monomers, sugar monomers is, that it's going to end somewhere with one sialic acid residue. Personally, I find this very, very confusing. I still find it very confusing. It makes me crazy. But there you go. So the GM series, one sialic acid residue. You have a GD series, disialic acid residue. So here you have two sialic acid residues, and it goes on like this. You have the GT series, you have the GQ series, mono, di, tri, quarto, quinto. It just goes on like that. Okay, so let's go ahead and take a look at a really, really important medical application of ganglioside biochemistry. So let me go ahead and go on to the next page here for this one. So let's see. Yes. So let's do this in red. So a particular medical application. Now, Tay-Sachs disease is caused by the accumulation of ganglioside GM2 in the brain and spleen. Okay, so lipids, these membrane lipids, the body breaks them down and replaces them on a regular basis. So it has mechanisms in place. There are specific enzymes that actually break them down, in this particular case, one sugar unit at a time, until it it ends up with ceramide. And then the cell takes that ceramide and goes ahead and builds another, another lipid with it, another ganglioside, say, something like that. Well, if there's something wrong with one of these enzymes and it can't actually break down, let's say ganglioside GM3 or GM1 or whatever it is that it can't break down, that it stops. The breakdown process stops, and that particular molecule at that step starts to build up in the cell. The buildup of GM2, ganglioside GM2, is what causes Tay-Sachs disease. So that's what's happening here. So let's take a look at this. Okay, brain and spleen. Okay, so Tay-Sachs disease is caused by the accumulation of ganglioside GM2 in the brain and spleen. Actually, let me continue this because the body lacks the enzyme, in this particular case, this enzyme, hexosaminidase A, which catalyzes the following metabolic step. And since we're talking about a breakdown, we're talking about a catabolic step. Catabolism, it's the breakdown of a molecule into simpler parts. Anabolism is the buildup, it's the biosynthesis. Metabolism is the general term, refers to both. So metabolic, I'll just say catabolic step. So the body lacks this particular enzyme. It's called hexosaminidase A, and because it lacks it, it cannot break it down past a certain point, and it builds up, and then all kinds of terrible things start to happen. And yes, Okay, so here is the particular metabolic step. So we have our ceramide. I'm just going to write it as our ceramide backbone. I'm just not going to write out the whole structure. Let me do this in blue. So we have our ceramide, and then we have a glucose residue, galactose, and then we have, let's go ahead and do one more here, and let's go ahead and connect one more here. So we have, in this particular case, this ganglioside has glucose, galactose, and it's a little bigger so I can actually write it in there. So boom, 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 boom. Gal, NAC, N-acetyl galactosamine, and of course we have our new 5-AC, our sialic acid. So in this particular case, so this is GM2, ganglioside, monoganglioside 2, that's the name, GM2. This particular molecule goes through the particular metabolic step. It's supposed to do this. Okay. I haven't drawn the oxygens in. I'm just using hexagons for the sugars. There's oxygens, of course, on here, but you know that already. So gal, NAC, so it's supposed to go to ceramide, gluc, gal, and of course we have our new 5-AC. Okay, so what's supposed to happen, the metabolic step, the hexosaminidase A, I'll go ahead and do this in red. Hexosaminidase A. The enzyme catalyzes removing that N-acetyl galactosamine residue off and takes it to this particular molecule is GM3. And of course this one goes on in further metabolic steps. We remove this one, we remove the galactose, we remove the glucose, and finally we end up with ceramide, and the ceramide can go and do what it does. If hexosaminidase is not available, if the body lacks this enzyme, this metabolic step can't take place. If it can't take place, this molecule ends up accumulating in the brain and spleen, and this is what causes Tay-Sachs disease. So that's what's happening. This is ganglioside, another ganglioside. This is GM2, GM3, there's GM1, GM whatever. And again, this M, it stands for one sialic acid residue. There is our sialic acid residue. There is our sialic acid residue. This 2 doesn't refer to the number of sialic acid residues. It just happens to be part of the name. That's it. It's one of those situations where they decided to go with a nice short name, GM1, GM2, GM3, GM4. That's all that's going on. Okay, now let's go ahead and talk about our last class, which is going to be the sterols. Okay, so the basic structure of a sterol, boy, I really need to start writing a little bit more slowly here. So the basic structure of a sterol consists of this ring system where you have these 4 rings that are actually fused together. So it's going to look something like this. See, make sure I give myself enough room here. So we have that, and we have this, and we have this, and we have that. Oops, what am I doing? That's not right. Well, it's right, but it's slightly off. It's actually going to be like that. And then we have this R group, and I'm going to go ahead and put this little OH group because it's either going to be a hydroxy or it's going to be a carbonyl, something like that. So this is the basic structure of a sterol, and we call this the A ring, the B ring, the C ring, and the D ring. So 6-membered, 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 and 5-membered. So now let me go ahead and just draw out what is probably the most important example of a sterol. Certainly the example that you are most familiar with. So important example. And this is going to be something called cholesterol, or you're more familiar with it as cholesterol. So that's it. There's the sterol part. That's the little prefix telling you what it does, what it is. So again, cholesterol, cholesterol. We just shifted the accent a little bit. So it's going to look something like this. So again, the basic structure is going to remain. Going to have a couple of changes. I'm going to have a particular R group. So let me make sure I have enough room here. So there's one. There. Let me make these a little bit better. I want to make it as pretty as possible. We're actually going to see an actual picture of it in just a moment, but it's always nice to draw them out, to be able to draw them out by hand. So that's that. Then let me see here. I've got a double bond here. Let me not forget my hydroxy group. I've got a CH3 here. I've got a CH3 here. I've got a C and a CH3, and I've got 1, 2, 3, 4, and 5. So CH3, CH3, this is a CH, this is CH2, this is CH2, this is CH2. So let me make sure I've got all my carbons, because whenever we start drawing these things by hand, there's always some atom that's missing. 1, 2, 3, 4, 5, 6. Yes, 1, 2, 3, 4, 5, 6. Yes. On this one, we have a CH3 branching off, CH3. Okay. If I've drawn it correctly, this is our important example of cholesterol. And of course, now we're going to take a look at some pictures of this. So cholesterol, this is cholesterol. And notice what we have. This actually shows the stereochemistry. So these H's, this one is out, this one is back, this one is back. This CH3 group is forward. This CH3 group is forward. I mean, it's coming out toward us. This is coming out toward us. This, not a problem. This OH group is coming out toward us. So again, It depends on what it is that you're talking about. If you're talking about something where stereochemistry is going to be important, where you're getting into the actual chemistry of the sterol, then yes, you're going to see something like this. If not, then you'll just see regular line structures. Okay, I decided to go ahead and bring back this particular illustration down at the bottom half of the page so you can see now that we've discussed all of the classes. So we have our sphingomyelins here. Let's see, so we have our sphingosine. So we have our C, our C, our C. So this is our sphingosine. This is the backbone of the sphingosine right here. And then we have, of course, the amide linkage, the R group. And in this particular case, we have the phospho group right there. And it looks like we've got choline right here. So that's an example of a sphingomyelin. Our cholesterol is this one. That's weird. Why did I put that extra? Anyway, so this is our cholesterol, and notice this is outer leaflet, this is inner leaflet. So cholesterol like that, and notice this, all of this non-polar. These are all just carbon-hydrogen bonds. This is just, hydrocarbon portion of it. It's the hydrocarbon portion that's actually down inside of it, but this OH is pointing towards the outside. You notice the cholesterol here, the inner leaflet, this is inside, and this, the outer, oops, go ahead. The OH group is actually pointing in towards the cell. It's actually interacting with the aqueous environment inside the cell. This OH group, the polar part of the molecule, is interacting with the aqueous environment outside the cell. So phosphatidylcholine, this is going to be one of our glycerophospholipids. Again, you have your long chain, you have your phosphate, and in this case it looks like you have the choline there. Okay, so now our inner leaflet, we have the phosphatidylserine, we have another cholesterol, phosphatidylethanolamine, phosphatidylinositol, just something else attached to it. So we have our phosphate, our phosphate, our phosphate. So that's it. This pretty much covers the discussion of our membrane lipids. Thank you so much for joining us here at educator.com. We'll see you next time. Bye-bye.